Welcome to episode 2 of See the Unseen.
I'm Suzanne.
And I'm Jussi.
We are postdocs at the Kavli Institute for Nanoscience in Oxford.
Today we will be talking about my research in the Vector Lab.
And what do you do there?
We study the cerebellum, or models of the cerebellum.
Okay, and what's the cerebellum?
The cerebellum is part of the brain. It's located at the back of the skull, and for the longest time we've known that it coordinates or controls our movements. When we talk about the cerebellum, there's usually a schematic of a basketball player throwing a hoop, and you can see him calculating the movements of his arms to get the right trajectory to get the ball in the hoop. And when the cerebellum doesn't function properly, when there's damage, or if there's for example a genetic disorder, you get what's called ataxia, which means a lack of coordination of motor and movements. And we really want to understand what causes ataxia, and if there are ways for us to ease the symptoms or even cure the disease.
So you're trying to understand how the disease happens and how to treat it.
So I will be taking you through some of the steps in the protocol, or the recipe for brain organoids.
Okay, so what's a brain organoid?
Oh, that's a good question. So organoid as a word means organ-like. So we're basically working with organ-like structures on a dish, and in this case they will be more brain-like.
Okay.
And specifically cerebellar organoids.
So where do you get these little mini pseudo-brains from?
You make them from stem cells.
Yeah.
So I can show you some of the stem cells as well, and then I can talk you through how to work with them to make the organoids.
You're going to teach me how to make a brain?
Yeah. So you can take any cell from the human body, and you can force them to express 4 different genes, and those genes will then reprogram or make that cell into a stem cell.
So I can take like a skin cell and an eye cell, and I do the same thing to them and they become the same kind of cell.
And it's really cool because we can use these stem cells to study how humans are different.
Okay, so once you have the stem cells that you've made from someone's skin, what do you do with them?
Well, you culture them for a bit. I can actually show you, I have some growing in the...
You've got some human stem cells in there?
Yeah, I do. So these were plated or put on this plastic yesterday, so they're still a little bit sparse, but as they grow they will kind of fill the whole well. And you can see that they're very tiny little cells, and they form these small regions of cells. We call them colonies of stem cells, and they will proliferate, they will grow and multiply on this well, and once the well is nearly full, you can take them and make an organoid. I started a new differentiation 2 days ago.
And by differentiation you mean?
Yeah, we call them differentiations, and it's a round of differentiation, taking the stem cells to how many days of differentiating into this organoid. Okay, so here we see what's going to become an organoid. So you have this halo of stem cells that are not being integrated into what's going to become the organoid. And in the middle you see a very well-defined blob of cells called the embryoid body. And that central bit is going to become the organoid.
When you showed us the stem cells earlier, they were growing kind of in a layer on the dish. And then here you say some of them are in a layer, and in the middle, are they making like a ball, or? Yeah,
that's not actually a layer. So it's a ball of cells, and obviously with this microscope we only see a 2D image of that ball.
Yeah.
Okay, so this is a new 96-well plate of a differentiation that I started a week ago. So they're looking at a little bit different. They've got rid of the halo of dead cells, and now all the dead cells are kind of spread all over the well. But you can see the well-defined blob of cells, the embryoid body, here. And with time and changing the medium and changing the plates, this will grow.
So how big will they get?
We'll see, because I have organoids growing, and it's day 80 7 today.
Yeah?
Yeah?
So when?
Yeah. Now? So the yellow bits, or slightly pinkish, maybe yellow bits, are the organoids. And we can't even see the whole organoid because of the field of view isn't big enough for this organoid. But you can see that it has a well-defined border, and it's starting to grow some of these internal structures.
So what do you mean by internal structures?
Well, some of this tissue will grow into these rosettes. I can actually show you images of the rosettes later, but they do tend to organize in a certain way within that organoid.
So there's a lot of nerve-like cells inside there.
Yeah.
Are they signaling to each other, or?
We know that in different types of brain organoids, people have shown that there's spontaneous electrical activity. We haven't yet looked at our organoids, but definitely in some brain organoids you do see electrical activity between cells.
Huh.
Like I said, in the lab, you can take images of these organoids, you can section them into very, very tiny, thin slices, and then you can color in the different types of proteins or the different types of cells that you have in them. And here you can see different kinds of colors and cells, and the green cells here are nerve cells.
Ooh.
And the blue color is just showing where the nucleus of the cell is. And when I mentioned the internal structures on the organoids, I mentioned the rosettes. So this is one rosette.
Oh, that's really pretty.
Yeah. So they tend to make these internal structures, and these are very like nerve-like structures, or neuronal tissue-like structures. You can also, so these are from an organoid that you cut through into thin slices, but you can also break up the whole organoid and then put those cells on a glass and do the same, so you color them in, and then you can image that with a microscope. And here the red color is again showing the nerve-like cells.
My favorite kind.
Yeah. And then the different colors, there's the blue again showing where the nucleus is.
So what are you doing with all these brain organoids?
Oh, that's a good question. There are different people working on the same organoids for different reasons. Like I said in the previous video, I'm a medical doctor, and my specialization is in child neurology, so I'm interested in developmental brain disorders and diseases.
So if you thought you had maybe a drug, like a potential drug that you thought would be good for this disease, instead of testing it in human patients and kids, you could test it
Exactly.
on these
Yeah.
little
Yeah.
mini brain organoids.
Yeah, that's the really cool part.
You're taking healthy stem cells and then you're mutating them bit by bit to try and make them mimic the disease.
Yeah.
And then once you have mimicked the disease, you'll be able to use that for drug screens.
Yeah, that's it. You got it.
And what are the next steps for you?
Well, I would like to go back to the clinic and continue working with patients and with these, what we call disease models. So that's our aim. We want to make a disease model from these organoids that kind of mirrors what's actually going on with the disease.
So you can take what you learn from the clinic and apply it to the organoids. You can take what you learn from the organoids and see how that works in the clinic.
Yeah, exactly.
So we are so excited now that we can just start asking anyone in the Kavli. We want to interview people about the wide range of science that goes on here, from DNA to discovery science.
Yeah, and I think Manish is next. And just at the end, we'd like to thank Esther and her lab, everyone who's helped us, and especially our funders.
And thanks to Jussi for sharing his science with us, and to everyone who's watching.